So let's now just kind of step through the different phases that we go through. And what do we want to learn at each of these phases? When we're in phase 1 and we're doing a simple phase 1 study, we want to make sure that we're developing a prototype formulation. We want to try to start to lock in on what are the ratio of excipients and API and finalize that this is the group of excipients that we want. We want to start to identify the API physical characteristics and chemical characteristics to start to understand how is this material going to behave. We want to look at the particle size of the material and the flow properties, and we're also trying to hone in on what dissolution profile we're looking at. So basically, by the end of phase 1, we pretty much want to have our prototype formulation selected. Going into phase 2, and at the start of phase 2, we're going to start up our scale-up process. And we need to scale it up to an appropriate scale. That is going to depend on what the processes we're looking at, what the drug is, how large we think the commercial batches are going to be. So there's a number of factors in there that will help you determine what scale you want to be at this point. And we want to start to look at the optimization of the process and the optimization of the formulation. We want to start to look at things like how much mag stearate do we need to add to this formulation? How much Avicil do we need to add to the formulation? How long do we need to blend, or how long do we need to wet granulate, and how much water we might need to add for the wet granulation. So by the end of this, we want to be able to finalize our quantitative formulation for each of the excipients, and we want to start to hone in on what are the in-process and release specifications that we're going to need, and what are the important parameters for our formulation. So by the time we get to phase 3, we want to have a scaled up formulation for the pivotal batch, which needs to be within 10% of what our commercial scale is going to be, or a minimum of 100,000 dosage units. And you want that very close to the final formulation and process, so that when you go to file your NDA, there's not a lot of questions about what you put into your pivotal bio batch and what actually is going to be marketed. You can make some final adjustments to, let's say, mag stearate by processing it a little bit, but you don't want to be making large-scale changes at this point. And the other thing you want to keep in mind at this level is you want to make sure that all of your your analytical methods are validated. This will help you later on in making sure that everything is hunky-dory when you go to file the NDA. At the same time, we're going to manufacture our registration stability batches. And what do we need for those batches is we want to make sure that we have the final shape, the final color, any printing or embossing that will be on the tablet. Now the printing doesn't have to be the exact logo that's going to end up being on the tablet when it's commercialized. It can just say 1, 2, 3, it can say XYZ. Same way with the embossing. It doesn't have to be the final embossing. It just should be on that tablet if you're going to have it for the commercial product. And as far as the packaging configurations, we need to know if we're going to be doing blisters, and if we're doing a blister, whether it's aluminum or a laminate in aluminum and so forth. And as far as bottles go, we need to know how small we need to go. Do we need to worry about doing physician samples of 5 to 10 in a bottle, or what is our lowest amount, and then what is the highest packaging configuration? The other thing we need to make sure we keep an eye on is the API synthesis should be finalized by now, and it should be very close to its final batch size. We don't want to see a lot of physical or chemical changes happening to the API at this stage of the game. The other thing we want to keep in mind about our rework procedure, or our transfer, or our scale-up, is rework procedures. We talked about roller compaction a little bit earlier. Again, if we know we're going to have fines and we want to reprocess those fines, we need to take that into account now at this stage. We also want to take into account any holding times for our materials. If we granulate a material, how long can we hold that before we compress it? Can it be a month? Can it be 2 months? That's something we want to look at. Or if we manufacture a coating solution, how long can that coating solution be held before we actually do the coating? And the goal at this point is that we're going to be ready for that NDA filing. And at the NDA filing and commercialization, we're really talking about everything's final. We've got the specification of the process, the finished products there. We've got our dissolution profile finalized, and we do now know what is the final print or embossing that's going to be on the tablets. We also have to take into account at this point validation batches. And if we do our scale up appropriately, the validation batches should go very smoothly. The question at this point is, normally companies would like to file the protocol for validation in their NDA, but they prefer not to execute those before the NDA is filed. The reason being is they would like to use those as their first 3 marketed batches. And because of this, they want to manufacture it as late as possible to keep as long of expiration date as possible. So our goal at this point is we want to pass that PAI when it comes up. We want to be ready for launch, and we want to be able to use SUPAC guidelines. If we have to make any changes, we can go by those guidelines.